The actual protein synthesis happens now, during translation. Mature mRNA leaves the nucleus and comes upon the rough ER, rough endoplasmatic reticulum, which is covered in ribosomes. Ribosomes are structures composed out of proteins and RNA themselves. Translation of the genetic info on the mRNA into a protein occurs in and through these ribosomes. But what are proteins? Proteins are chains of different amino acids. The mRNA now enters with its 5' first into a ribosome. Inside the ribosome are 3 slots. Each of these slots has room for 3 bases. Every slot has a name and a specific function. The first one is the A site, amino acid site. The middle slot is the P site, peptidyl site, and on the left is the E site, or exit site. 9 bases in total. Imagine it like a conveyor belt tunnel. Groups of 3 bases are called codons, and every codon represents an amino acid. The start of translation does not happen randomly. There is a universal rule, which is that AUG is the so-called start codon. Translation begins when AUG is in the first slot, the A site. From now on, every codon, a group of 3 bases, codes for the protein. But how are amino acids which make up the protein linked to codons? The amino acids get delivered to the ribosome with mRNA in it by tRNA. T stands for transport. tRNA is a construction with 3 bases and 1 amino acid at its top. This enters the ribosome in the A slot, and the 3 bases then complementarily link to those of the mRNA. Now the ribosome moves along 5' to 3' and the mRNA in the other direction, until the bound pair is in the middle or P site. A new tRNA can bind in the A site, bringing its corresponding amino acid with it. This state of ribosome is called pre-translational. Now the tRNA, which is in the P or middle site, transfers its amino acid onto the amino acid bound to the tRNA in the A site. Ribosome and mRNA move along. This state now is called post-translational. Now, simultaneously, the tRNA in the E site leaves, exit, and a new tRNA joins the A site, recreating the pre-translational state. This process continues and a chain of amino acids gets formed. Translation happens until the ribosome comes upon a stop codon, of which there are 3: UAA, UAG, and UGA. When one of these stop codons enters the A site of the ribosome, translation and thereby protein synthesis is finished. The ribosome and the tRNA disconnect from the mRNA. Afterwards, the amino acid chain, or protein, disconnects from the ribosome and is thereby finished. We now have made a protein using the copy of a gene.